Welcome to Inert Chemistry. In today's video lecture, we will discuss an important topic, that is deamination reactions of amino acids and their importance. Let's begin the lecture. What is deamination? In the previous video, we started the degradation of amino acids. As you know that the degradation of amino acid consists of two parts. The degradation of amino group and degradation of carbon skeleton. The degradation of amino group is further classified into transamination, deamination, transport of ammonia, and at the last urea cycle. The first step that we had already learned is transamination. During transamination, the transfer of amino group from an amino acid to keto acid takes place. As a result of this transfer, another amino acid, glutamate, is formed. This glutamate acts as a collecting center of amino group from various amino acids. Note that glutamate is the only amino acid which undergo oxidative deamination. So most of the amino acids during transamination form glutamate. Then this second step is deamination. During the deamination, the nitrogen atom or the amino group present in amino acid is removed from amino acids as ammoniamine. Note that during transamination, there is only transfer of amino group from an amino acid to keto acid. But during the deamination, the nitrogen atom of amino acids is removed from amino acids as ammoniamine. Deamination is of two types: oxidative deamination and non-oxidative deamination. Oxidative deamination is catalyzed by glutamate dehydrogenase enzyme or another enzyme, that is amino acid oxidase. While non-oxidative deamination is catalyzed by amino acid dehydratases. During the oxidative deamination, deamination is coupled with oxidation. While in non-oxidative deamination, oxidation do not takes place, but only there is deamination. Now first we see the oxidative deamination by glutamate dehydrogenase. Here an amino acid glutamate is shown, and as you know that glutamate acts as a collecting center of amino group from various amino acids. L-dehydrogenase enzyme acted on the glutamate and first formed an unstable intermediate alpha amino glutarate. This alpha amino glutarate is further decomposes and form alpha ketoglutarate and free ammonium ion. This reaction is freely reversible reaction, and this reaction functions both in amino acid catabolism and biosynthesis. For example, catabolically, it channels the nitrogen from glutamate to ammonia, means that during the catabolism, the reaction takes in the forward direction and glutamate is broken down into alpha ketoglutarate and free ammonium ion. While anabolically, during the biosynthesis of amino acids, the reaction proceeds backward and alpha ketoglutarate and ammonium ion recombines to form glutamate. This glutamate dehydrogenase enzyme is located in mitochondria of liver cells. It contains zinc and having 6 identical units. It is allosterically regulated and inhibited by GTP and ATP and activated by GDP and ADP. Steroid and thyroid hormones inhibit this enzyme. It is the only enzyme that can either use NAD plus or NADP plus. This is responsible for most of the ammonia formed in the body. This enzyme reversibly links the glutamate metabolism to TCA cycle. As you know that when glutamate is broken down, two products are formed, the free ammonia and alpha ketoglutarate. And alpha ketoglutarate is one of the intermediates of TCA cycle. So we can say that this reaction reversibly links glutamate to TCA cycle. This enzyme is involved in both catabolic and anabolic reactions of amino acids. Oxidative deamination by L-amino acids. Both L and D amino acid oxidases occur in the kidney and liver. Amino acid oxidase. Amino acid oxidases use auto-oxidizable FMN or FAD as coenzymes. For example, L-amino acid oxidases use FMN as coenzyme, while D-amino acid oxidases use FAD as coenzyme. In liver and kidney, the L-amino acid oxidases can convert an amino acid to alpha amino acid, an unstable intermediate. This unstable intermediate is decomposed to alpha keto acid and release the free ammonium ion. The reduced FMNH2 is reoxidized by molecular oxygen and forms hydrogen peroxide. This hydrogen peroxide is broken down into water and oxygen by catalysis. D amino acids. D amino acids are not present in human bodies. They must be taken through diet and metabolized within the body. The D amino acids are converted into respective alpha keto acids by the action of D amino acid oxidases and free ammonium ion is released. D amino acid oxidases use FAD as coenzyme. The alpha keto acid forms, they may either convert it into L amino acids by transaminases or they may be used as an energy source or act as precursors for the synthesis of glucose and fats. Now let's come to non-oxidative deamination. During the non-oxidative deamination, there is no oxidation. Some amino acids do not undergo oxidation, but they are only deaminated. For example, serine, threonine, and homoserine. They are acted upon by amino acid dehydratases and converted into their respective alpha keto acids and release ammonia as NH4 or ammonium ion. Similarly, cysteine and homocysteine, the sulfur containing amino acids, they are acted upon by the amino acid disulfurases, sulfur hydrases, and converted into their respective alpha keto acids with the release of ammonium ion. Similarly, there is deamination of histidine to urokinase. This is an example of non-oxidative deamination during which histidase enzyme is used and it converts histidine to urokinase and release free ammonium ion. This is all about deamination. Thanks for watching this video. In the next video we will see that how to write this topic as a long question in exams. Please like, share and subscribe the channel.